Thanks a lot to the EBI team for the kind invitation here. It's our pleasure, I have to say, to present to you today the journey to fair bioimage data management. It's not only me, but oops, I have to see how I can switch forward. Not only me presenting, but also my colleagues Tom Boissonnet and Christian Schmidt. Tom and me, we are located in Heinrich-Heine University in Düsseldorf, and Christian Schmidt works at the DKFZ in Heidelberg. But all together, we are also part of the big project NFDI for Bioimage. You can see the logo here in the right corner. And we are also part of another project I will introduce to you in a few minutes. Okay, let's get started. So the outline of our journey today is that I will give you a little bit of the introduction where the journey of imaging data starts. Let's do a little bit of a reality check, and then I would like to introduce to you the two projects I mentioned already and how they can support you on the journey to fair imaging data. And then Tom will take over with the practical part. He will give you a day-to-day image data management introduction to OMERO. That's the tool we focus in one of the projects a lot on. And then Christian, at the end, once you have your imaging data under management and in fair conditions, he will show you how you can refine them, how you can reuse imaging data, and that can be fun and hopefully has really high impact for your science and your research projects. So let's start in the beginning. Where does the journey start? So usually the journey starts if you want to acquire imaging data behind the microscope, right? So in a situation like that. So in Düsseldorf, in my place, in our place, it means you might come to our imaging core facility. So I'm the director of the imaging core facility in Düsseldorf, which is the Center for Advanced Imaging, or CAI. Please check out if you're interested in what we are doing. And then you start to acquire images. Great. And where does the story end? The journey ends. So hopefully it ends with a wonderful publication, and maybe your imaging data are so impressive that you are invited to design even the cover for one of those high-ranking journals. Yeah, and that is then maybe the whole life cycle, but many, many other things happen in between. And that's really what we have to talk about today, because you need really a lot of tools and services and expert knowledge to do really good bioimage data management and to get your data in a fair state. So, and you heard about all these aspects during the webinar. I guess many of those things had been covered, and we try to do a little bit of a wrap up today. And as mentioned by Patricia, a little bit of showing you a few tools we could offer to you and where we could support you to use them. So what is important, of course? It's not only image acquisition, but of course, storage is very important, and how you access your data is important. Then you will process and analyze the data, prepare them for the publication, and then it's about archiving and long-term storage, making the data public. So for example, you could upload your imaging data to Bioimage Archive, which has been introduced as well, of course. And of course, we as a local provider for support also collaborate with people with a team of Bioimage Archive. So this is the theory, yeah. And this is really mentioning all the different things that are important. But now let's maybe have a look to the reality. So how does it look there in reality? And this is displayed very nicely in this cartoon-like description of the life cycle of imaging data. And maybe you know it, right? If you do go for image acquisition. So yeah, somehow I will remember the laser power. And usually I always take the same value. If not, I can take some notes, maybe like that, right? And then we move on. So just storing the image data in the PC at the microscope, nobody will delete them, or am I right? And for the analysis stuff, yeah, there's one script that everybody's using in the lab. There was this very skilled postdoc. We always use the same. That's fine. For the publication, oh, you have to really describe what you did. Okay, what can I remember what I did there? Still, the data is on the PC. And we just need a stick or an external drive to transfer them. And then in the very end, if you, for example, are going to upload this for Bioimage Archive, they ask you for the naming, and then you have something like that, yeah. So that's in German. So day 1, day 2 imaging, day 3, day 4. There's yeast, even one important information about the data, and so on and so forth. So what do I want to show you? That there's really a big need for tools, for services, and for experts, because in many places, this is the reality, how it looks like if you work with imaging data. So there is really a need for funded projects to improve the situation, to make data fair, to make data reusable, to make scientific data reproducible as well. And this has been recognized by the funders, luckily, not only in Germany, but in also many other places. And that's why we are in the lucky situation that we have a funded project running still, which is called the I3D Biobyte Project Information Infrastructure for Bioimage Data. And I really invite you to check out our websites, because this tool is really focusing on the practical aspects. So we focus a lot on OMERO, which will be introduced by Tom. So how you deploy this image data management system in your institution. You can work together with us. You can find on our websites a lot of teaching materials and also recording of our events. And we make those teaching materials reusable for you. This is shown in the next slide. So this is the team. And I really would like to mention that these are the PIs, so who wrote the application. And those are the people you really need, those experts who work on, like Tom, on the deployment of OMERO and on producing all this very, very excellent training materials like Christian did. And this is really important to push this extra activity of image data management or research data management in general forward. And you can find here under the training materials a lot of nice resources. And I really invite you to check out what is maybe suits you or what kind of information might help you in the future. Okay, so the next project is a little bit bigger. It's called NFDI for Bioimage, and this is a consortium of the National Research Data Infrastructure. Now in Germany, there was the idea we would need really a big infrastructure for all the different disciplines in research and science. And so let's set up a bottom-up project which takes care of data management in general. So, and our part is we take care of all steps of research data lifecycle for microscopy and bioimage analysis. So this project is really broader. It's much bigger than I3D Bio, and it's part of the bigger overall infrastructure we try to set up. So the main objectives are a little bit broader within NFDI for bioimage. It's really about standardization of the bioimage data type, which is still a problem because, we deal with many proprietary formats here. And this is also important. We do not focus only on local infrastructures, but we really aim to provide federated, scalable infrastructures for fair image data. And as you can imagine, we also collaborate for this aspect with the Bioimage Archive team at EMBL-EBI, because this could be the endpoint for many of the data we want to publish, to make available those data and have really a pipeline from our local infrastructures into this environment. The third very important aspect is about analysis, because as I mentioned in my reality check slide, the reproducibility of especially analysis workflows is always a big problem. And here we really want to maximize the reach of reproducible analysis workflows, which are available for many people, for example, in the cloud. And last but not least, and I think that's most important, that we really capacitate the researchers, that they know what they want to do. And that's why we really love to be here today, to give you some information about this kind of activities and tools we can offer to you. And this is all part of NFDI in general, so the National Research Data Infrastructure, which has the vision data as a common good for excellent research. So all data, all disciplines organized by the scientific community in Germany. And that's really a very interesting challenge, because it's very bottom up. So we work together with 26 consortia, and they cover all scientific disciplines. It's not us only from life science, it's from humanities, it's really cultural science, it's from the engineers. So very interesting to work together with all of them. And it's really an investment into people and not into methods. So it's not that we have much storage provided by this project, but we really have many data stewards, many research software engineers, and so on. So we are one of the 3 calls with our consortium. We are part of it. So in the end, now we have 26 consortia for disciplines and 1 cross consortia project. We started 2 years ago. And the good message is that we have a long-term perspective for the whole thing. This is the data steward team in general. And for example, those people could help you if you need support to prepare your images for publication in a public repository like the Bioimage archive. So, or maybe because you come from a different country and there's a similar initiative, they would forward your request to this people because they are really well connected with the community in general. Okay, so now I need to find the next slide. So, and this is already my last slide because what is, as I mentioned, super important that this is really cross-discipline activity, and also cross-discipline support is now ensured by working together, of course, as you can imagine, mainly with the NFDI consortia from the life science field, what is presented in this slide. So there are many other consortia. We work together to give you the best support we can give. And with this, I am at the end of my part, and I hand over to Tom for the practical things. So, and I will maybe stop here.
And I can take over. Okay, you see my screen now?
Perfect.
Okay. So on my side, I want to talk a bit more about the practical aspect on how you would conduct data management day to day in your lab. But the first point I like to mention always is that the data management should be for everyone working. So including the group leaders, the experimenters, and in third, the bioimage analyst. And by this, I mean that the group leaders are the person having the overview of all the projects. They are the one who needs also to monitor the progress of the different projects. So have a look at the incoming data that experimenters are acquiring, but also they need to care about long-term archiving. So this topic of data management is really also important for group leaders. In second, I list the experimenters. So they're the person who knows all the details about the different projects. They would know which experiment worked, which had an issue, and that they take notes to remember. So in their lab notebooks, either in physical form or electronic. And so they are the ones who are able to do the data organization, also the data annotation, so that other people in the lab know what has been going on the bench. And the bioimage analysts, so they would need this metadata for the processing, so some information they might need to run the analysis pipeline. But they also need a way to share the results back. So you process some results, you want to give that back to the experimenters or to the group leaders, so for everyone to also see what the processed data looks like. And you need to have some kind of traceability of your analysis. So say you find value X at the end of the analysis and you see some outliers. Well, you would like to come back to the image and see where those outliers come from. Okay. But because we're also talking about image data management, it should also be very important that everyone has an easy way to see the image. And this is not necessarily trivial. So if you worked with microscopes before, those are measuring device. We work with different dimension in our images. So one of them are the channels in the image. This is a typical image you could get from a microscope in these 3 different channels. But you assemble this channel to make color. But if you miss the metadata, so there's always the metadata coming with the images, then you wouldn't know what were the different channels for these images in the end, or the most important information, the pixel size of the images. Different dimensions again, talking about stacks or talking about time series. So this is what it would look like for this stack or another time series here. But a distinction you have to know when you're doing imaging is that an image and a file can be different things. So most of the time you would get for one file, one image inside. Still with imaging, we may have image size range, a broad range for the image, for the file size going for a few megabyte to terabyte. Or you could have the situation where in one file you have n images, so some kind of archives that microscope can produce, or the other way around that you would need 1,000 files to encode a single image. In the case of Z stacks or time series, this can be very common. But really what we don't want to be doing is file management. What we want to be doing is image data management because this is our single experiments. So again, we need an easy way to see the images. And about file formats, often a question would be how to open the file that produced that contains the image, because we have all different file formats. One common solution is Fiji, and if you have used Fiji before or opened image with Fiji before, you may have seen this window before. And if I zoom in here, this is actually something called bioformats. And that's the thing in Fiji that is responsible to, well, allow you to open all different kind of file formats. And this is really a tedious job that Bioformats has taken of supporting a lot of microscopy file formats. Currently, I think it's around 160. And yeah, they are able to read all the data and the metadata to also interpret the pixel size metadata for all different kind of file formats, for example. Okay, but finally coming to the tool that we're interested in. If you have seen OMERO before, this is what it looks like. This is a web interface, meaning that you would log in into your browser to your server address, and you would interact with your data through this web browser application. But the data lies in the back on the server, and this is the one where you would upload your files. And of course, in between, you would find bioformats there so that we don't have to do any more file data management, but we are just caring about the image data from our end with OMERO Web. So OMERO is first a way to explore your data. So you have on the left here the list of all data sets that are acquired for a certain project. You get some preview in the center that you can have the preview of the image to know which one you want to select. And on the right, you would always have metadata appearing. That can be the channel names, or that can be pixel size or the name of the image, etc. But a button I really like to show is this one that would show you the link to the selected image. And that becomes very useful because now what you can do in a group, instead of sharing images, sending images to each other in different ways, you can have your data uploaded to a single OMERO server, everybody logged in into that server. And now sharing the links, people would see the same resources. So only sharing that thing, everybody knows what we're talking about. And so our data sets that we have on the left, those are all the experiments I conduct, and I have only these levels of organization for the data that are the projects and data sets containing the images. But if we need more to organize data, OMERO also provide support in the form of tags. And the tags are really the flexible thing that will help you organize your data sets. So really, in my opinion, they would be the Swiss knife of data organization. So I don't know if you've ever used tags for something before, but with those, you could emulate any kind of folder-like structure. For example, you could imagine that you create a single tag for any kind of folder you would like to create. So if you're used to organize your data on your computer, the rule of thumb for me would say that every folder I usually like to make, every folder level I make in my data organization, that could become one tag. The good thing is that you don't need to think of a hierarchy between the tags. You can browse them in any order. So for example, here, this project data is from a master module. So we were thinking how would our student like to find that data? And that's why we organized it in this tags, also putting them in some tag collection, this tag set. So for the group, and looking in the tag group 1, this would be all my data sets for the group 1. Or I could have the same for group 2 and group 3. But now if I'm more interested in the category of the lesson my students had, and I want to check the data sets for the lesson that every group has acquired, I could filter them like that. And so the tags, really allow you to filter your data, create your own data filters. And to give you more examples of how you could use them. So if you're doing training AI and you want to categorize your image in training, testing, validation sets, for the training of your network, you could create tags for those three categories and assign them randomly to images. That avoids any kind of copy of the data. The images always stay in their data set. We are not reorganizing data sets or projects or anything. We just assign tags and then I can filter the data to find by these tags. Or another example I would have is to say, I want to select a subset of images to show that I have to discuss with my supervisor. So again, select those images, discuss about those images, share a link to the tag even. And when you don't need the tag anymore, you could simply delete it. And this will not impact your organization of data at all. Another annotation we have in OMERO are the key value pairs. So this is the place where you can really go on describing your data. The hardest part with this would be to come with the template you'd like. I think Christian is discussing further on that, but finding the right information you want to assign to your different data sets, you could put that, on your data sets. And once you have that, OMERO provides some easy ways to copy paste from one data set to another. So once you have the template, you could simply copy paste it on all new incoming data. And if you need more advanced way of reorganizing this metadata, there would also be some scripts provided by OMERO. So there's also a way to add scripts to a server. And with this one, you could export all your annotation to Excel, modify them, re-import them, and so really have all the workflow needed to annotate data. But if you had to try a tool in OMERO, if you're not convinced yet that this may be useful for you, the selling point of OMERO would be this plugin called OMERO Figure that allows you to very quickly create figures out of your images. So you would put them into this, again, web application, organize your different panels, and add the metadata on top of your images, because what happens here is that the images are again loaded from the server, but it's not only the pixel values, there's also all the metadata coming with. So that could be the pixel size, for example, in this histology image. Or for this fluorescence microscopy image, we would also have the channel names displayed here, and that is coming again from OMERO, that someone has indicated what were the name of the different channels of the image, or it's read from the metadata. And very rapidly, you could create those panels. The nice thing about OMERO figure is that once you have finished your figure, it's still alive. So by that, I mean, the link to the original data is still preserved. You still have the link to the metadata. If you look on the right, you can also see a scroller. I would also like the laser pointer. There's also a scroller here to go through the Z stack for the different panels here. or you would have to scroll to the time series in here. So the image still come from the server and is still linked to the metadata. But also, you still have a link to the figure so that you could also send that figure to someone. You don't have to make it a PDF necessarily. You could directly send the link and someone could play with that data. Okay. The last point I want to make concern more people than that would do data analysis. And to the server, we connected before with OMERO Web to look at our data, play with our data. But the server, you can also connect with different programs. So Fiji has a plugin for OMERO. You have QPass with a plugin, or directly with Python code. You could directly connect to the server and get the data out of it. Also metadata. So OMERO has a database in the back, and you can really get information in any shape out of the server. So if I try to put all that together, that means someone at the lab would acquire images, upload them to my OMERO server, growing the size of my database. This is not a file anymore. That becomes an image then in OMERO. Uploading more images, I increase the size of my experiment, organizing them in projects and data sets, adding some annotation as key value pairs or as tags to organize, make different view. Maybe my supervisor helped me doing this job. And would come a bioimage analyst pulling out data in Fiji, for example, performing image analysis, obtaining measurements and ROI. Those you would also upload to OMERO. And someone could then finally do the job of taking this data out, putting it in a large data frame, or if you want a large Excel file to create your plots that you would put on your paper. So I want to mention we are not. I'm not doing a full tutorial on OMERO right now. If you're interested in more content on how to use OMERO, you could use, for example, YouTube. We have a series with i3D training material there that you could use. But if you simply type OMERO, I'm sure you would find a lot of very interesting resources. So to conclude, I want to say that again, this is really for everyone in a project. So your group leaders, the experimenters, bioimage analysts, everyone has something to take from that tool. And it's really there to help you explore your data. So you have a remote access to the data. You can share links over files. You have a traceability from the data acquisition to analysis, and you can then use developed extension that help you again interact even more with your data. So something like OMERO Figure. And that would be it for me. So I would then let Christian continue.
Thank you. So you should be able to see my screen already. And yeah, I'm taking over from here. I'm Christian at the DKFZ in Heidelberg. And as you see on the slide as well, I'm a scientific project manager. So I'm not as deep in those topics as like Tom, for example, is. But I like to have that perspective because I like to refer to myself as the ordinary biologist because that's my basic background. I've been working in immunobiology for quite some time. So I want to give a perspective on what we've learned. From this seminar today, but also from the seminar series and the previous slides, and take a little bit like where is the journey right now? Where are we heading at, and where are we already? And for that, I start with the introduction slide that Mike Smith gave here. He talked a lot about why research data management is so important, and one of the things was, are you able to identify the data also after some time? Are you able to find your own data or someone else's data that you need? And if we now think of published data, and sometimes when you read a paper, you might be very interested in more specifics and testing out, for example, an analysis workflow with the data that's already there to see if that is suitable for you to reproduce in a similar way with your experiments, then we are probably very much familiar with the typical situation that somewhere at the end of the paper just says the data is available upon request, but trying to request that data often fails, unfortunately, for many different reasons. And those things that we've been discussing about fair data management, they're really bringing us forward to improving that situation. And here is an example. And that would be now from a collaboration that we had with people in Dresden in the i3D Bio project, together with whom we established their local OMERO instance. Now, this would be a publication that they had. This is not marine or environmental ecology related. This is about pericardial graft models. So they analyzed a lot here with tissue samples. But let's say I find this figure and now I'm interested in the details. I want to examine for whatever a little part of the figure that I'm specifically interested in. And the classical situation where I only have the images as the representation in JPEG included in a manuscript, I can basically do nothing with the image. Any useful information is lost, it's low resolution, so I'm depending on getting access. And in this particular example, they used what Tom has just been speaking about, because they have now an OMERO instance, and with the link from the publication, you're able to find the OMERO-based figure that they have done here. And that gives you really a big advantage by having the access to the original data underlying the figure. So in this case, I can just click on the figure that I'm interested in or in any of those and see here through the web client that is here for this OMERO instance, what the original image is. And now you're already familiar with this view. That's what Tom just showed. You have access to their public part of the OMERO instance, find the original image, and also find the attached metadata to see, for example, to which treatment groups this belonged, and so on. So I have now the ability to really find more details than could be included in just the publication figure. And I have access to the original figure, right? I could even download this original data now in high resolution, reanalyze that, use it with QPath, whatever I want to do. So this already is quite a cool advancement that I can make data available like this for others to reuse, but it's probably not the end of the story yet, because this is not, for example, with a persistent identifier in OMERO. To find the data, you need the link from the publication. But it's a very good way of using that tool already to make data more alive out there. And the greatest thing here as well is that you're also allowed clearly to reuse the data because there is a permissive license attached with the publication and the data. What also you saw, and Tom has highlighted this, is that the data is enriched with metadata, and Tom explained the tags for helping to organize the data and the key value pairs. And as he has mentioned, the REMBI checklist. So REMBI annotated metadata, as you would find, for example, here, to clearly say what is the recipient biological entity that received the pericardium graft here. And I'm now looking a little bit again at this, even though this has been explained before, because for the bioimaging world, if you focus on the imaging part, REMBI is really very important to keep in mind. So it is made in mind with the question of what information do I need as a researcher to understand what I see and what has been done with an image. And therefore, you have different modules and components that you can also hierarchically put onto your data, generally describe your study, and include these important informations like what are the authors, who are the authors, and what is the license, and what is it in general that has been done here? What is maybe a link to the publication that has been set? You may have several study components with different imaging methods included, for which you can then individually describe the modules that you find here that go around the bioimage data lifecycle, include information on image acquisition and image data that could also wrap, for example, if there has been something done in the area of image analysis workflows, so the analysis data, and so on. So this is really helpful, and I recommend looking at the original paper. But you might find that this paper is not the go-to guide on its own already. And that is really because also the authors said here, this is a community endeavor. They agreed, they said, we want to distinguish recommended metadata requirements from any particular computer model in which that is wrapped. So this is only about what annotation is needed. But it doesn't dictate to you in which format it is annotated or how exactly you use that in your electronic lab notebook, in any Excel file, or if you should do it on OMERO. But since we've been speaking about OMERO, that is something I don't need to go into detail right now, but I recommend to you lots of training material that we have done, including the one that Tom has already also shown today, the general i3D Bio training material. There's lots of workshops that we conducted, and upon request, we are also always happy to extend that to other locations. So have a look at these recommendations for how to do Rambi annotation in OMERO as an option. But I want to pick up at one point because it's also very important, and we have found that it's often a hurdle for researchers to talk about ontologies and what that actually means. So I'd like to repeat at a high level, so to say, what is the relevance of ontologies? And that would be something that is recommended in Rambi. So if we look, for example, at the study component module of Rambi, then we see it gives you the imaging method and the study component description as keys that you should describe. And here it also says use where possible ontologies to describe that. So what does it mean and why would I use that? And for that, I'm giving you an example from the field I've been working in. Say I have annotated my data nicely with key value pairs, and I've worked with CD4 T cells in my work. So I say here, that's a cell type, it's CD4 positive T cells, and I'm working on a disease model called experimental autoimmune encephalomyelitis. Well, someone else talking about the same things might not talk of a cell type, or maybe a type of cell, or the cellular entity, or the cellular identity. And the same goes true for the actual value. So is it a CD4 plus T cell, or is it a CD4 positive T lymphocyte, or is it even more specifically a naive CD4 positive T cell, etc. So that there are different ways of how I could describe my data. And that gives us the challenge that we need to avoid ambiguity, that we need to be objective in the description, that others can also understand that, and it clearly describes what it is. And on top, if we have metadata that we also want to use with the computer to access that, then it should be machine interpretable. And that's really where ontologies come into play. So what I would hope you remember after the talk is ontologies, what are they? They're a conceptual framework of how you grasp the knowledge in a specific scientific domain, the things that we have in our heads and in the textbooks, and then represent that in a way that the computer can read it. And in order to do so, the individual terms, the vocabulary of those domains is attributed with properties and relationships of the terms. There are specific groupings into classes of the individual term instances that it has, and you have different ontologies in different research domains, and they can be also mapped to each other. So maybe you want to describe a specific cell in the context of all the cells that you know, or you want to describe it in the physiology of the immune system. And if there are two ontologies, they can be mapped so that this can be adopted. Of course, domain knowledge is extending. That's why we do research. So they are evolving over time, and that is why those ontologies are typically versioned. And this is very important. They are formalized. So those ontologies can be expressed in a specific ontology format that a computer can read. And here are just examples listed. An example would be if we have these cells again, I have a representation of a respective domain knowledge. For example, if I'm working with T cells, I know that any T cell is a lymphocyte, and lymphocytes belong to the leukocytes, and so on. So I have a hierarchical clustering that is described in a way that I can also represent as a graph, which can be helpful for people doing data analysis to represent knowledge graphs. And that really gives a great advantage if we use those individual terms. Because let's say I take a single key value pair just from an ontology. And in my case, the example was a CD4 positive alpha beta T cell, which you find here in the tree of the EFO ontology. Then implicitly in that term, I have included much more information than I have actually said in the individual term, because the ontology has formalized that is a cell that's carrying, for example, alpha-beta chains, not a gamma-delta T cell. It is a mature T cell, so it has completed thymic selection. It's circulatory properly. It's not sticking in the thymus. It belongs to the hematopoietic system, etc., etc. So these are things that if I work in immunology, I know, of course, but how would the computer know that? And of course, there is large language models that can grasp now very much information from context, but to be really specific and to make that programmable without having the black box of what is in the back end of trained models, ontologies really help to make the computer know what is behind those knowledges. So that's just what ontologies are, and we recommend in the training material that you indicate ontologies as you have seen on the slides from Tom, for example, that you use the term in its natural language for humans, but then also the accession numbers of the ontologies. And we do that because it's in line with the ISA framework, which is pretty well established. And if now people ask us, so do I now need all of my terms in ontology compliance? What a huge overload. We typically say, if you would have to pick 5 or 10 most important keywords that describe your data, then make them compliant with ontologies at least. And that already brings you quite a step ahead. This is for later when you review the slides. There are some links for typical ontologies in bioimaging. But what I like to spend the last couple of minutes with is picking up what we've seen before, and let's get more concrete on what we can do now. So these are the take-home messages from the presentation of Stéphane Pesson earlier in this webinar series. And if I pick that up for bioimaging data, I can go through my checklist. That really helps me to make sure I know the things that we need to think of. So I'm working with bioimaging data. That might be some light microscopy or electron microscopy techniques. I will use the RAMBI checklist as a starting point for the metadata regarding my bioimaging experiments. Then, if available, I can do that metadata curation, for example, in OMERO's key value pairs. I can think of the way of how I will collect that metadata and organize it ahead of my experiments. I know that there are file formats that are suitable, and Tom has just talked about the bioformats and the compatible bioformats compatible proprietary file formats, but you can also use the open ones, OMITIF and OMIZAR, coming back to that in a second. So we should use bioimaging ontologies plus potentially domain-specific ontologies. And as this webinar series is originating from the area of marine biology and ecology, and I'm not an expert in that, I just picked one up. And here is the environment ontology, from which I just first Googled marine plankton, and I have no idea of these things. So I found dinoflagellates as a term. So I looked that up in the environment ontology, looking, is there actually representation? So it's represented with the term dinophytae. There is the synonym dinoflagellates in that spelling also included. So that's a term that is ontology compliant and has a synonym. Maybe I want to use that to look for some data. You might remember the talk by Ibuki from the Bioimage Archive, who talked about the potency of having a central public Bioimage archive. So let's try that. And for that, I just typed in those terms, dinophytae and dinoflagellates, in the search term of the Bioimage Archive. And interestingly, when looking for the recently released data sets, I actually found one from the area of this webinar, from the TRACK consortium. So that's actually only released last week. Lucky shot here, I guess. And if we look into details of this data set, I find a lot of information that is really helpful for me to first assess what I have here. So I could find my keyword back. That's why the data was findable in the archive. And then I can look more into detail on what I have. It's ultrastructure expansion microscopy data from large culture collections of marine plankton. And if I look at the file list, I not only find myriads of files of imaging data in different sizes with RAMBI compliant annotations. But what's really cool about this specific data set is I even find an S3 bucket in which the data is contained, so an object store. And I find an access point to a MOBI project. And for those who don't know what that is, let's first think back, what is OMIZAR again? And here I might remind you of the talk by Josh, who has explained the Bioformats 101. And in essence, it's a chunked and cloud-ready file format, or file container, however you want to call it, that enables you to access data directly in the cloud without the need of downloading large volumes of data. So with that in mind, I said, let's try to access the data. And since I was on my private computer without Fiji on it, I just used a remote desktop. And this is something that's also been worked on from the NFDI for Bioimage Consortium in collaboration with many partners. BAND is the bioimage analysis desktop. And this is running on resources in Tübingen, which is the German network of bioinformatics, DENBI, and they provide cloud infrastructure in kind for NFDI. So here I could just spin up a desktop and remotely access the desktop from my computer to then access this desktop and do my image analysis there. And it has Fiji pre-installed, as you would expect in a bioimage analysis desktop. So I have the Mobi plugin in Fiji. I can just copy in this Mobi project link that was provided in the paper, and voila, I'm able to see here the list of collections of images that they have. And now to know what that is, I just need to find the metadata about it. So I went to the original paper that's linked from the Bioimage archive, and they have nicely a supplementary table with here a list of what the different culture numbers contain. So if I look, for example, at this image, I know now it's Mamiella gilva, even though I don't know specifically what that is. But this really allows me now to know what I'm looking at and to actually stream the data, the original image data from the S3 bucket in the cloud live to my computer, and then make my adjustments, my settings, and if you will, now you can continue with image analysis and reuse. So you really have access now to that. Again, there is the license to it. I can reuse it. Okay, that just as a brief show, I think to summarize, we are really getting closer to fair bioimage data sharing. There is still things to overcome. The technologies dynamically evolve. For example, spatial omics data management is still quite challenging in many aspects. There are people working on that also in the NFDI for Bioimage Consortium, and we will also put that in focus in i3D Bio if continued funding. And there is now really lots of guidance for you as scientists or as core facility staff where to start. There is detailed checklists on how to do measurements with microscopes and quality checks from Crowd-Blimi. You've seen the RAMBI checklist. There are publication guidelines, what to put in your figures and how to publish data. And the most important thing is you're not alone. There are all the projects that have been now shown to you, and there is people who really professionalize in this and are super willing and friendly to help out there. And with that, I'm a little bit over time, but I would like to thank briefly all the different people. So this is really a large community endeavor internationally with many projects and many people working together to imagine or imaging the future. And with that, I would like to thank you for your attention and I think open the question round.
Thank you very much, Stephanie, Tom, Christian. And also, as you can see, our amazing speakers have already answered many of your questions, but there are still two remaining. The first one is for Tom, actually. It says, the spatial genomics domain is generating quite a lot of imaging data as well. Do you think OMERO can support the modality as well?
So just before I answer this question, I answered some of the question, but if someone also wants more development on the answer I gave, we can also discuss about that. So to that, I would say, so with OMERO, you could support a lot of things, but the workflows are not necessarily here. So it would probably need some work to integrate your spatial genomic images inside OMERO, and how do you link to the rest of the genomic data. This is what we want to be working on. So adding a partner to the i3D Bio with an expertise on spatial genomics data. So to improve the workflows to have the data in OMERO as well. That's the answer I would give. It is possible, and we have a lot of mechanisms to do things, but if it's for programmers only, this is not working, you could say. So simplifying those workflows.
Thanks a lot. Next question is open. How do you enforce user compliance with RAMBI, etc.?
So maybe that points rather in my direction, because I think there is many things you can do. So of course, in the end, we want to see our colleagues and the scientists and researchers that they really understand and see the benefit. And this is actually what Christian aimed for, to show how powerful it is if you have applied REMBI to your data. They will be reused not only by you, but maybe also by others. And then you have really, hopefully, real impact in this kind of sharing your data. So that is really what is the vision in the end for NFDI, for all of these projects in the beginning, to make it work, because that is maybe a little bit of a far goal. So it is very helpful if you are, for example, in a bigger research project, like in Germany, we have the CRCs, this different, maybe 10 groups, 20 groups working on one topic, and then they have agreed on a data policy where they all agreed to use or to apply really fair image data management, fair data management in general. And then it is just mandatory for those people who are part of the project to use those things. So it's a little bit of a carrot and a stick thingy. So in the end, there's a benefit, but in the beginning, to make it work, you can enforce a bit by having this kind of policies in place. And there it really helps to work get started with a smaller subunit of people working together on one topic, which makes it, even from the practical side, much easier to handle, because then you can have a template, what has been described by Tom, which really fits for all of those people working on the same topic. And so you have to see how you can make it easy for the people, support them, having a little bit of enforcement by the policies, and then in the end they will hopefully do it themselves because they see the benefit. Maybe that's what I would answer to that.
Thank you. I'm gonna jump a bit to the last question because it's related to REMBI. When do you introduce the concepts of REMBI, etc., before or after the microscope introduction? So principal investigators need to be on board.
So what we do in our core facility, so that's really local in Düsseldorf, but I know it's similar in other places, is that people get microscopy introduction, and then they are invited also to get the introduction to OMERO and to have a meeting with one of the local data stewards and then they are introduced to REMBI. Yeah, that is, so it's not yet mandatory to do it. People have the choice still. They don't have the choice if they are already part of one of what I mentioned, this bigger local projects. Yeah, then they have to do it. But at the moment, it's not mandatory, but it's very, very close to the microscopy introduction. Yeah. And so what helps a lot is if you do it with students. So we do it in our master student courses. They get the introduction and then they start freshly from the beginning. They never do it differently. And so that is really the easiest and the best investment you can do, because they just do it and they think one has to do it just from the beginning. And then the good thing is they immediately see the benefit. Yeah. They don't have still data somewhere. They now have all to annotate. Yeah, after 2 years of acquisition, you maybe not so much in favor for doing annotations for this tons of data. But if you have a fresh start, then that's really easy.
Thanks a lot. Next question is, what is a major pain point in spatial data standardization?
Yeah, so there are several pain points. One of them is that this field is also technology-wise expanding and evolving so quickly that even some of the vendors don't have really all the metadata that you would want to have from the research side included. So, for example, here at the DKFZ, Jan-Philipp Malm, who's heading the single-cell open lab, is also in contact a lot with these people about the formats that you have. Then spatial biology or spatial data is also not only one technology. There are basically two major ways of how that is done, and there are myriads of individual little pieces of how the techniques really work. There is some starting, I might copy some links here to this question. There is some some starting discussions about that. And there is now the European Society for Spatial Biology dealing a lot with where spatial omics goes. And a subgroup of that, headed actually by Philip Malm through the Task Area 3 in NFDI for Bioimage, is the so-called IO Fast initiative, in which people work on how to do the data management with spatial omics. So there is a lot of development, and if you are interested in more details, that is the contact that I will share here in the answer.
Thanks a lot. I'm aware that we're very close to ending. Do you mind if we take the last 3 questions? Yes.
Sure.
Okay. So, next question is there any relevance of bioimaging in plant biology?
Yes, we especially, for example, in Düsseldorf, we have a big cluster, excellence cluster for plant biology. And so we do a lot of plant imaging. Yes. And we do have templates available for how to annotate your plant images, let's say, right? Yeah, sure.
Thanks a lot. Next question is, can I access images stored in OMERO through third-party software?
So that I can answer. There was a similar question also raised earlier about accessing the results of IMARIS. So OMERO by default hides the file you upload to the users, but there's, well, ways you could expose those files. Another way is to import data with something called in-place imports. That is having data on a file server and having this file server, well, linked to the OMERO server. And so both OMERO then is able to read your images, but you could also access data on that file server from any third-party software. And you could then store results next to your image files or import the results again to OMERO. So it doesn't change how OMERO looks at images. It's just change where the data is coming from, so from a file server. And so this would need to be set up by your administrators. So that's not something the user can make themselves. But if you know your administrators and you're interested in that, you could, well, first come to the forum image.sc to also ask questions, or invite your administrators to go there, and they would find the support from the community. And I can paste a link to this in-place imports as answer. Yes.
Thanks a lot. And last question, could you expand on the role of data stewards in this?
So this is a new role in our universe, which is super important to make all those things happen, right? So really you need this extra capacity you have with the data steward team to support people to find their way through ontologies. As Christian explained, you really have to know where are those resources, yeah? So it's not part of our daily training for our PhD students and within the research projects. And we now are within those projects are really setting up all these kind of materials and this kind of infrastructures. The teams, the different data steward teams from different disciplines have to be linked to each other. That's a very important role in the whole research data management business, not only for imaging, but for all of those activities. And so that's actually a new profile of how you could work in scientific environment, right? So it's very important to have those people. And that's why NFDI is really investing in people and not into like hardware, computing resources. It's really those people who make the whole thing work, yeah.
Got it. Thanks a lot. Just one comment from our participants. Thank you all so much. This was very informative. Great presentations and mind-opening information. Are there any closing remarks that you would like to give?
Thank you very much for everyone who was here today, and please don't hesitate to reach out to us. We're really for collaborating and helping and bringing things together.
Yeah, same thing from my side. So join the community with ImageSC. That's probably the best way to get in contact with someone. Yeah.
Have a look to the websites. Just get into contact to the data steward teams. They're really willing to help you.